The movement of water and ions in plants is a fundamental process that sustains life, supports growth, and regulates physiological functions. Water acts as the medium for the transport of dissolved nutrients and ions from the soil to different plant organs. Essential ions such as nitrate NO, phosphate Po cubed, potassium K, calcium Ca², magnesium Mg, iron Fe², Fe cubed, and trace elements move through the plant via complex physical and biochemical pathways. Understanding these mechanisms is critical for MPhil students because it forms the foundation of plant physiology, nutrient management, and sustainable agricultural practices. Water and ion movement is tightly coupled and occurs at multiple scales from the soil-root interface, through root tissues, and via the xylem and phloem, to leaves and reproductive organs. Advanced understanding requires integrating plant anatomy, biophysics, membrane transport, and environmental interactions. Water enters plant roots primarily through osmosis, driven by a water potential gradient between the soil and root cells. The water potential Psi is influenced by solute concentration osmotic potential, turgor pressure potential, and metric potential in the soil. Water flows from regions of higher water potential soil to lower water potential root cells. There are three main pathways of water movement through roots. Apoplastic pathway. Water moves through the cell walls and intercellular spaces, bypassing the cytoplasm. This pathway allows rapid water flow but is blocked at the Casparian strip in the endodermis, forcing water to enter the symplast to reach the xylem. Symplastic pathway. Water moves from cell to cell via plasmadsmata, allowing selective transport of ions and molecules along with water. Transmembrane pathway. Water passes through cell membranes and vacuoles, often facilitated by aquaporins, specialized water channel proteins that regulate hydraulic conductivity. Once water reaches the xylem, it is transported upward to leaves and other organs through transpiration-driven bulk flow. This is known as the cohesion-tension theory, where water molecules are pulled upward due to transpiration at the leaf surface, cohesion between water molecules, and adhesion to xylem walls. Environmental factors such as light, temperature, humidity, and soil moisture strongly influence transpiration rates and, consequently, water movement in plants. Ion movement in plants is more complex than water movement because ions carry charges and interact with plant membranes and soil colloids. There are two primary mechanisms for ion uptake. Passive transport. Ions move along electrochemical gradients without the expenditure of energy. This is typical for ions with high concentration in the soil relative to the root cytoplasm, such as nitrate NO under fertile conditions. Active transport. Ions are transported against their electrochemical gradient using membrane transport proteins, such as ATPases and ion-specific channels. Active transport is crucial for essential ions like potassium, calcium, magnesium, and micronutrients that are often in low concentrations in soil. Once inside the root cells, ions are loaded into the xylem for upward transport. Xylem loading involves both apoplastic and symplastic pathways, sometimes aided by transfer cells with high surface area for active ion exchange. Ionic transport in xylem sap is largely mass flow, driven by the transpiration stream, although some diffusion occurs for specific ions. Xylem transport. Water and ions move from roots to shoots and leaves via xylem vessels and tracheids. Hydraulic conductance of the xylem determines the efficiency of water and ion transport. Essential macronutrients such as nitrate, potassium, and calcium are transported in ionic form dissolved in the xylem sap. Xylem transport is unidirectional root-give-shoot and influenced by transpiration pull, root pressure, and soil water availability. Phloem transport. While xylem primarily moves water and minerals, phloem transports photosynthate sugars, hormones, and some ions to various sink tissues. Source-to-sink movement relies on pressure flow mechanism as flow driven by osmotic pressure differences. Certain ions like potassium can regulate phloem loading and unloading by maintaining turgor pressure in sieve elements. Ion movement across membranes is regulated by selective transporters, channels, and pumps. ATPases create electrochemical gradients that drive secondary transport of cations and anions. Potassium channels facilitate K uptake in root hairs and guard cells for stomatal regulation. Calcium channels act as signaling elements, mediating responses to environmental stress and nutrient deficiency. Aquaporins regulate water permeability, allowing plants to respond dynamically to soil moisture and osmotic stress. The rhizosphere, the soil region influenced by roots, plays a critical role in water and ion movement. Root exudates, including organic acids and enzymes, alter soil pH and solubility of nutrients. Mycorrhizal associations extend the effective root surface area, enhancing water and phosphate uptake. Soil microorganisms in the rhizosphere influence the availability of nitrogen, phosphorus, and micronutrients, indirectly affecting ion transport. Water and ion transport is influenced by soil moisture and hydraulic conductivity. Dry soils reduce water availability, limiting mass flow of ions. Temperature affects transpiration rate, membrane fluidity, and enzyme activity for active transport. Salinity. High soil salinity reduces water potential, leading to osmotic stress and altered ion uptake. Nutrient concentration. Deficiency or excess of specific ions modifies uptake kinetics through feedback regulation. Plant developmental stage. Young leaves and reproductive tissues act as strong sinks, drawing water and nutrients preferentially. Optimizing irrigation strategies requires knowledge of transpiration rates, hydraulic conductivity, and root architecture. Fertilizer management relies on understanding ion uptake kinetics, mobility in soil, and Xylem, Phloem transport patterns. Biofortification strategies target enhanced uptake and translocation of micronutrients to edible tissues. Salinity and drought studies focus on water use efficiency, aquaporin regulation, and osmotic adjustment mechanisms. The movement of water and ions in plants is a complex, integrated process governed by physical laws, membrane physiology, soil-plant interactions, and environmental conditions. For MPhil students, mastering these processes is critical for research in plant physiology and nutrient management, supervision of experiments on water stress, nutrient transport, and crop improvement, advanced understanding of agricultural sustainability and soil-plant water relations. By combining knowledge of soil-water dynamics, Xylem and Phloem transport, membrane transporters, and rhizosphere interactions, students can predict, manipulate, and optimize water and nutrient delivery for enhanced plant growth and productivity.